Hi, good to see you. Oh, one last time. I thought I was done recording these things, but um, just as a teacher, I want to do this one last thing, and it's to help you learn things in lab. And let me explain what this is for. It's partly because of the loss of the 3 weeks that we had in lab and the unfortunate result of that, which is that the material that you were learning in lecture and lab are offset from each other. You were learning things in lab that you had learned about in lecture weeks before instead of them being kind of more side by side, which is how I've designed the course, and I like it that way. And I didn't like that I couldn't have it that way, this semester. And another thing is needing to eliminate material. And I eliminated material out of the central nervous system chapter. So while I'm having you learn central nervous system stuff in lab for the lab practical, I'm just not able to have it be something that I lecture about. But I also don't like having you learn anatomy that doesn't seem to have meaning behind it. And so that's why I'm going to do this. So I'm just, making it optional. But if you want to have the anatomy that you're learning for this last practical be more meaningful to you, then this will help. And I think it will just also help with some of your other classes. Like, I don't feel so bad eliminating nervous system stuff since we have so many neurobiology courses and we have neurobiology faculty here. So you can kind of get this stuff elsewhere. But maybe you don't get the anatomy as much as you do maybe the neurophysiology and some of the cellular specifics of neurons and things. But so I'm going to talk a little bit about that. Another thing to note is that I made the videos for this last practical months ago. I made them in August, I believe. It was either late August or early September. So I recorded them before we had that 3 week gap, and I recorded them with the idea that we would have had more time to do all these things in lab. And so there's more anatomy that is pointed to in the videos than what you're responsible for. And so just like always, make sure you're using those handouts. It's just 2 this time. Make sure you're using those handouts and the list of terms that are on those handouts. And that's the realm of the material, even though there are extra terms that are in the videos because I made them so long ago. I was just trying to make sure I had enough time to do things. I couldn't imagine if I hadn't made those earlier, all the amount of work that it would have been. Anyway, so with that said, I am going to forge ahead here and just give you some information about these bits of anatomy. So last time, here's my laser pointer. And so just some basic regions here. When you're taking a look at the sagittal sections of the sheep brains, you can notice, these really obvious structures. The cerebrum, which you also see from the other side, looking at the brain externally, that is this whole area that has all the wrinkles in it. I'll talk about the wrinkles in a little bit. Cerebellum, this large structure here, which is quite separate to the cerebrum. There's a crease in between it. And this thalamus, which you think of as the kind of the top of the brainstem and a major part of the midbrain. And what I want to do is just kind of go up the brain. I'm taking slides that I would have used, but I eliminated some text from it just to simplify things. But then I was realizing, yeah, I don't have a lot of time. So I didn't do a lot of adjustments to the slides. And so on some of these slides, not this one, there'll be excess text. Just ignore it. So with the brainstem, we learned in lab some of these structures. We learned medulla and we learned pons, and then midbrain, like superior colliculus, inferior colliculus, those structures. Those are these midbrain structures along with the thalamus. Even though when you look at them, they're very, very white, there are these areas of gray matter within them. And I told you about white matter versus gray matter in those lectures. Just remember that gray matter are the areas where there's cell bodies and synapses, there's things happening there. And then white matter is just things passing by, just axons going by, transmitting information from one place to another. So whenever you see gray matter in an area, then that means, well, there's some sort of function to that spot. There's something going on there. And so let me just talk a little bit about the gray matter areas that are in the medulla. The medulla, at the very beginning of the brain, has these centers within it. It's called reticular formation. I actually took that out. And those are centers for very, very basic control of autonomic functions. So parasympathetic, sympathetic, control of heart rate, make it go up or down, things like that. Now, the passageways out will be different, going down to vagus nerve, for instance, for parasympathetic, or coming out of the thoracic spinal nerves and cervical spinal nerves for sympathetic. So some of the anatomy of the descending signals end up being different. And then there's a vomiting center. I kind of like talking about that. I won't go into those details. But there are some important descending tracts. So it would be white matter that's a part of the medulla oblongata. But these tracts, they're called pyramidal tracts because when you cut them in cross section, it looks like a pyramid. And these are very just major tracts for voluntary movements. So information traveling from the brain to control your body crosses over at the medulla to get down to the spinal nerves where they'll actually go out and control your muscles. The pons just above that, when we look at it in the sheep brain, it's like this kind of little bulby, kind of stick-outy little spot, pretty obvious. And there's a lot of crossover, but it's tracks from communicating with the nearby cerebellum. And so that's why it kind of swells out there. And then there's also areas of the reticular formation. And reticular formation is not just in the pons, it's actually all along in this area here, but that sends signals to the cerebrum, just general excitability. So not specific signals for sensory information, but just to enhance the activity of the cerebrum. And so that happens to basically wake you up. And so the reticular formation gets information from like sounds, things you see, like you could be sleeping, but a bright white light would wake you up, and that's the reticular formation waking you up from that input, or sounds, things like that. I'm talking about, there's all these different kinds of input. Okay, and then going up to the midbrain, this is where we saw those interesting structures that look like a butt on a toilet seat. Remember me talking about that in the lab video? So that's a superior colliculus and inferior colliculus. And this is where they are. The superior colliculus is associated with vision information, and the inferior is associated with auditory information. Now, it's not where vision is perceived, but the superior colliculus is important in being able to have this kind of quick back and forth with the optic nerve. And so instead of having to go up to the cerebrum to do this and then go out again, it's this kind of quick back and forth at the midbrain for tracking things. So to be able to track objects. And so that's what this image is talking about. They're showing the optic nerve and the crossover. That's another thing that we see in the sheep brain. We do see the optic nerve, we see the crossover, we see the optic chiasm. And we see information coming in here. Now, information from the optic nerve is obviously going to go up to your cerebrum, be perceived as this visual information, but there's also this information that goes to the superior colliculus, and then you can see this information. These are efferent signals out to the eye muscles. That's where I say motor, and that's in red. And so that's to track things. And that's an interesting thing to know, that it's this lower brain function for tracking. I actually have sort of a sad medical story about this. It's and it comes from, I think it was in the 90s. There was the somewhat controversial case of this woman that was declared brain dead because of a lack of electrical activity in her cerebrum. The controversy was that the hospital and doctors were recommending that she be taken off life support. And I think the life support was feeding her. That's why it was controversial. And the family did not want to do this. And so it was in the news. The government is getting involved, I believe. But, and I can't remember her name just because it was 20 years ago or more. And anyway, I guess the name is less important as what the situation was. And so the situation was her family could bring a balloon in the room and she could track it. And so she had that ability. She had no cerebral activity. She had no evidence of cerebral activity. But the family was taking the fact that she could track these things as evidence of brain function, of higher brain function. But it was midbrain stuff. But, what can you say to families in those situations? They're holding on to their loved ones. Okay. So and I'm realizing that the picking up from my screen is not perfect. I'm trying to do this pretty fast, and I'm just trying to have it being good. But I realize my slide is not. You know what? Maybe I'll just pause it and see if I can fix that. Okay. Yeah, it wasn't really a big problem. Oh, actually, wait a minute. Just a second. Okay, there we go. Fixed it. Okay. Now onward to the cerebellum, and that's an interesting structure, and that there's a lot of communication when you have voluntary movement. There's a lot of communication in the cerebrum, but then there's communication to the cerebellum. Now, the cerebellum is not a place that initiates motor movements. If you have damage to your cerebellum, you'll still be able to move, but you won't have as much coordination. And, maybe I keep wanting to teach you things. Maybe I'll try to insert a movie in here that shows. There's always these cute little videos somewhat cute, of these cats. You don't want to laugh at them, but these cats and dogs that have hypoplasia of their cerebellum running around. It helps to coordinate motor movements, to smooth movements. It's also the area where you can store motor movements. And so sometimes people talk about motor memory. It's called procedural memory. But learning skills to be able to do something like swing a golf club, hit a ball, hit a baseball with a bat, and all those kinds of motions that you need to practice, like you hold your elbow up and you the way that you move your wrist and swing through. And so there's all these things that you need to remember that you practice and practice and practice, but then they become automatic. Well, what's that? What's becoming automatic? Why does practicing make your movements more improved? And so it's because of these procedural memories that are happening in your cerebellum. Um, and you know what? Maybe I'll see if I can try to insert it here. Okay. Play, you stupid thing. Okay. This. Okay. This is a video of the cat with cerebellar hypoplasia. I think there's two, both cats have this, um, and uh, oh God, um, they they actually acquire it. I think it comes from a virus that infects either when they're very, very young or infects the mother and she gives birth to cats like this. I, I forget the details of it. If I were lecturing normally, I'd look up all this stuff, but uh, I don't have I don't have time to do this. But I know that it has something to do with some viral interaction, and I believe it's during development, so that it would be some sort of viral infection of the mom. Oh, okay. So if you ever want to watch more of this, just look, go to YouTube and look up cerebellar hypoplasia. Okay. And then for the thalamus, it's that really real obvious structure right there in the middle at the top of the brainstem. And it's a pretty easy structure to identify in lab. Oh, just a second. Okay. Um, so this image I think is really great to understand what the thalamus does. And the thalamus is sort of like Grand Central Station, some big organizer and director of information that's coming up from the body to direct it to its proper place in the cerebrum. And with that, it's also a little bit about controlling how you can pay attention to particular stimuli, that you filter out unwanted stimuli and you are focused on stimuli that's important. And it's a really important structure in regards to keeping focus. And it's something that I think about a lot because I see my son having issues exactly in this area. He has Down syndrome. He's somewhere on the autism spectrum. He has Tourette syndrome. And I know when and his problems just really arose when he went into puberty. So about 3 years ago, we started to see him really decline and have effects with Tourette syndrome, and which he didn't have before. And he was able to run and have pay attention to things, and he was like a more typical kid with Down syndrome before that. And then with the onset of this Tourette's, the Tourette's has influences around in these same areas. There's an area that I'm not going to lecture about, but it's called the basal ganglia, and they are close by, and there's communication between them and the thalamus. And he just has focus issues, and it's really impacted his life. And I'm hoping at some point that maybe, because I know Tourette's often goes is less when you mature and become more of an adult. I'm hoping that he'll be able to regain his focus and have less of those effects. Um, okay. Oh, and the way that they did this image is through a dissection. So there's all these very interesting ways to do dissections, and these dissections are showing the white matter connections. And those white matter connections are also very important. We're starting to focus more on those rather than just knowing what the gray matter areas do. Moving up the brain, nearby the thalamus is the hypothalamus, hypothalamus, so just below the thalamus, makes sense, right? And the pituitary. And those are dangling down below. And you can see on the whole brain, half brain, you can see both of these. I'm tired of all the notifications. Okay. Hypothalamus and pituitary communicate with each other to be able to be the master control over the endocrine system. And so it's this neural connection to the hormone releasing section, which is basically what the pituitary is. There's a lot of vascularization around the pituitary. That's what this area is showing. And so the hormones that get left off from the pituitary travel into the blood, and then they can travel maybe to other glands and influence them, or maybe those hormones have particular effects. The hypothalamus has one of the more complicated connections of all the areas of the brain. It gets a lot of different kinds of inputs. It can influence your biological clock, and the information that is just basically that there's light coming from the retina, that goes to the hypothalamus, influences biological clock. So there's connections to the pineal there, and release of melatonin and things. Oh, you know what? I have the next slide is talks about the pineal. The hypothalamus is an area that controls your body temperature, your basic metabolic rate. So it's an important area. Then, and then I'll just talk about the pineal, which is also nearby. That's that kind of really obvious structure, just maybe a bit superior and posterior to the thalamus when you're looking in the sheep brain. It's a interesting structure. Has a endocrine function. It does release melatonin, but only at night. So it ends up being an important chemical signal to your body about for timing purposes, to be able so that your body knows kind of what time of day it is. And there's lots of biological processes that rely on timing. Your release of thyroid hormone happens in the morning. Well, how does your thyroid know that it's morning? It doesn't have a window into the world. So it receives a chemical signal in regards to that. And the cerebrum, of course, very much on the top of things there. Really big in humans, still big in the sheep. Mammals have big cerebrums. So big that in order to have more brain matter for your cerebrum requires this wrinkling. I'll explain that on my next slide. But there's these two main hemispheres. There's the fissure in between them. And then this is a dissection that where they kind of chop down a bit of the top of the cerebrum, and you can see this corpus callosum. That structure that we're also learning in the sheep brain lab. That's a white matter structure of lots of axons that are communicating, allow for the communication between the right and the left hemispheres. Sensory input, like from your body, what's called somatosensory input, so touch and temperature, those kinds of feelings, those travel like from your left side, it will go to your right hemisphere, and from your right side of your body to your left hemisphere. I'm actually reversing it on my own self. It's not right. But, and there's communication between the hemispheres because there is what's called lateralization of function. There are some functions that are controlled only in one hemisphere of the brain versus the other. I'm probably talking too much about stuff. This is taking a look at the cerebrum, seeing the folding that takes place. This is taking a look at the layering. So if you could take a look, a very close look, at the way the cells are structured within the cerebrum, there is this important layering process, this layering organization, and these layers need to be held intact. So you can't just slap down more brain and have more cerebrum. You have to have these 6 layers. So in order to increase the amount of neurons and in the cerebrum, you have to kind of add them in, but then have increased wrinkling. And, just like kind of like if you were trying to have more rug, but that's a stupid. Well, okay. If you want to add more rug, you can't, but the room is only so big. You have to just wrinkle up your rug. I don't know if that's a great allegory, a comparison or not. Okay. And then the cerebrum has different parts to it. These are just very basic areas. They match up with the bones that are nearby in the skull that you would have already learned: frontal, parietal, temporal, occipital. They're also laboring the cerebellum and the brainstem here, but there are those basic regions and the functions that are associated with them. That ties in with what I talked about regarding stroke, so that if you have a blockage and it's one of the cerebral arteries, and we learned about the anterior, middle, and posterior cerebral arteries, and then those are different areas, and then there's different functions that are associated with them. So you can see the functions that are listed there. Okay, I'm going to stop.